Let's jump right into today's explainer and uncover the fascinating, totally hidden physiological cost your body pays when turning dietary protein into usable energy. We usually think of calories in equaling energy out, basically assuming our food is just this generic fuel. But today, we're looking under the hood. We're going to see what really happens when your body treats dietary amino acids, the fundamental building blocks of protein, as a primary energy source instead of just structural components.
So we're starting off with a massive mystery today by asking a really fundamental question. Is the ultimate energy your body gets from protein-derived glucose exactly the same as what it gets from carbohydrates? Now, you might logically assume that a glucose molecule is, well, just a glucose molecule. Once it's circulating in your system, keeping your brain firing and your muscles moving, you'd think its energetic value is identical regardless of where it came from. But as we know, biology is rarely that simple. The origin story of that glucose actually changes absolutely everything about its efficiency.
And here's the crucial point. Glucose derived from amino acids actually yields a massive 32% less net energy than glucose from carbohydrate sources, and we are going to find out exactly why. Just think about that number for a second. Almost a third of the potential energy is simply missing compared to processing carbs. I mean, if you were running a business and almost a third of your gross profit just vanished into administrative fees, you'd want to audit the books immediately to find the leak, right? Well, that's exactly what we're going to do with your cellular metabolism today.
Okay, moving to our first section, the prep work, liver processing, and nitrogen.
To solve our missing energy mystery, we first need to look at the initial prep work that happens when dietary amino acids arrive at your body's main processing plant, the liver. When you consume them, they cannot just be burned for energy right out of the box. Nope. Step 1, they have to be metabolized by specific enzymes inside the liver. Then, step 2, and this is where things get complicated, the nitrogen has to be completely removed from them via the urea cycle. It's only after that nitrogen is stripped away that we reach step 3, where we're finally left with the carbon skeletons, which are also known as keto acids. So as you can see, it's a highly demanding breakdown process just to get the raw materials ready.
Now, these remaining carbon skeletons sit at a biological crossroads with two potential paths. They can either be used to produce acetyl-CoA, or they can be used to produce glucose through a specific pathway called gluconeogenesis. Both of these paths are absolutely crucial to cellular function. But for our energy mystery today, we are going to focus entirely on that first point, the intricate creation of glucose.
Which brings us to our next section, the transformation, alanine's path to glucose.
Before we track our specific amino acid, let's quickly define this intimidating biological term, gluconeogenesis. It literally just means the metabolic process of creating new glucose from those remaining carbon skeletons right inside the liver. Basically, your liver is manufacturing its very own new sugar supply, out of protein remnants. Let's watch this happen step by step with our hero for today, alanine. We start with the amino acid alanine circulating in the bloodstream, which gets picked up by the liver. Next, it meets an enzyme called alanine transaminase, which converts it into pyruvate. But the assembly line doesn't stop there. That pyruvate is then converted into oxaloacetate. From there, it travels through the remaining intricate steps of gluconeogenesis until, boom, it successfully emerges as a fully formed molecule of glucose.
But once that glucose is finally manufactured, the liver doesn't actually keep it. The newly produced glucose is released directly back into the bloodstream. The entire purpose of this elaborate manufacturing process, from alanine all the way to oxaloacetate, is just so that other tissues and organs in your body can grab this newly minted glucose and use it for energy.
But here's the catch, section 3, the final bill, the hidden metabolic tax.
And here is where we solve our initial mystery. Because in biology, no highly structured process is ever completely free. It requires a staggering consumption of 12 ATPs, which, as you know, is your body's fundamental energetic currency, just to synthesize a single molecule of glucose from an amino acid carbon skeleton. Think back to that conversion of pyruvate to glucose we just walked through. Your liver had to actively spend 12 ATPs just to force that manufacturing process to happen. It's the old saying, it takes money to make money. Well, in your cells, it literally takes energy to make usable energy.
Because of that steep 12 ATP consumption upfront in the liver, the net energy outcome is severely impacted. When this amino acid-derived glucose is ultimately oxidized all the way down to CO2, it only yields 26 ATPs. But get this, the metabolism of glucose derived straight from carbohydrate sources, that yields 32% more energy. That heavy, upfront manufacturing cost of 12 ATPs perfectly explains the exact 32% energy deficit we talked about at the very beginning of this explainer. You are paying a literal metabolic tax to force your body to turn protein into sugar.
So, I want to leave you with this final, provocative thought today. Knowing about this steep physiological tax, how will it change the way you view the actual usable energy on your plate? If your body is breaking down amino acids to create glucose, it is working so much harder for significantly less net energetic output compared to just processing carbohydrates. It kind of completely reframes how we think about the efficiency of our fuel, doesn't it? Think deeply about that metabolic tax the next time you're planning your energy sources, and as always, keep questioning the incredible mechanisms going on inside you